Novo assembly.
Hello everybody. I'd like to thank first the organisers for inviting me. It's a great honour to speak for an audience like this. So my title today is as Gil said, reference-free analysis about genetic variation. And this is all joint work with Gil and Mary, who was going to speak yesterday. And this is the outline of what we're going to do. So I'll start with talking about what are the reasons why we might be interested in doing. I sort of need this now.
For those watching at home.
Brilliant. Okay, so we start with why are we interested? What are the reasons why we might not have a reference, or we might not trust our reference, or might not want to use it? And then, so Gil was keen for me to explain sort of what are the current challenges and what are the real problems we're sort of facing. And so in order to do that, I'm going to start by describing the things we can confidently do, and sort of give you a sort of overview of the things that we know we can do quite confidently. And then once we're in this world where we can do reference-free analysis of, in fact, multiple individuals simultaneously, some new questions arise, and with the sort of new kind of scientific questions we can sort of ask and answer. And so I'll describe those, and then those lead us into sort of some new challenges and some new types of problem. So having started off explaining what we can do, I'll move on to the things that we find really hard, and having scared you with how hard things are, I'll give you an example of how we can still do something even in a really hard case. So why are we interested? There are many reasons why you might want to do things reference-free. You might be working on a species that has no high-quality reference, and you just may not be able to afford getting one. I mean, it's an expensive business. If you have a species that's got high heterozygosity or high numbers of repeats, it's not easy to build a reference. You might have some experimental situation. You have many divergent strains of a single species, and you want to compare them, and you might introduce artifacts by comparing them all with a single reference that is sort of diverged from each of them. So some people have already spoken about mapping biases that you have to deal with when you're dealing with mapping to a reference. And certainly when you want sort of a detailed understanding of a population, you might start worrying about novel sequence that's just not in your reference and that you're missing, and you don't have any measure of, how much stuff are you missing. And finally, the reference is just one individual, and depending on your species and how your reference was built, that's just somebody in some subpopulation, and it's never claimed that would have all of the sequence that exists in your population, and maybe you want to understand everything that's in your whole population. Okay, so that's why we're going to start asking these questions. So basically, I'm heading towards talking about de novo assembly. So what can we do? We can do diploid de novo assembly of variants. We can do it of multiple genomes simultaneously. That includes eukaryotes, including humans. And I'll describe that in a second. So we can call SNPs and indels inversions larger structural variants. And we've been doing this in the 1,000 Genomes project. There's been experimental validation of variants at different length scales, and so we're quite happy with what we've got. So that's great. We can study that for individuals. We can study that for multiple individuals. But at another level, what we can do is just use our methods to study the nature of the genome itself. And I'll give some examples of that. And now that we're talking about doing multiple people at the same time, or multiple populations, or whatever, we shall come to that. We can sort of simultaneously look at assemblies at the same time of different people at the same site, and maybe we can sort of transfer information across from one person to another. So how are we going to do this? We're going to do this with these things called De Bruijn graphs, which have been around for about 15 years. And I'm sure you've heard of them. They've been used in a haploid context. So generally you're trying to build a haploid reference mosaic genome that represents a diploid individual. I'm quite diploid oriented in this talk. And that's fine, but that's not really what I want to talk about. I want to talk about how you can use these graphs as a way of encoding polymorphism and repeats. So I'm just going to give a quick cartoon of how you might build one of these things. Essentially, the idea is that you're looking at a particular length scale at your genome. You've got a word size, call it K, and then you draw a sliding window across your sequences and watch the words that appear. And words that appear consecutively are joined together by edges in a graph. So on the top example up there, I've got an example of a SNP. And as your sliding window moved across the top chromosome, it would trace out one path, and as it moved across the bottom one, you trace out another path. And so a SNP creates this thing that we call a bubble. And the bottom example is the thing that confounds us. So those are two different situations which both could generate the same graph. At the top, you've got a C and G deletion, which generates a bubble, but alternatively, that CG deletion could be a paralog. Yeah, I mean, there's no way for your sliding window to know it's just local information. And so you've got two types of event that can generate the same graph. And a lot of the things we're dealing with are sort of trying to separate out these confounding events. So we have some software called Cortex. It's been around for over a year now. So the first thing that we had to deal with is the fact that de novo assembly is very memory hungry, especially with new sequencing data. So if you wanted to assemble a human genome with something like SOAP de novo, that might cost you 512 gigs of RAM. With Velvet, it would be even more. So we spent a long time developing a very memory efficient hash table in which we could encode the graph. And so for the low coverage, the high coverage individual inside the 1000 Genomes project, we managed to build all of the Illumina data in just 64 gigs of RAM. Now, that's a best case scenario. I think the data was quite clean, and if I included 454 data, suddenly my memory requirements go up. So 128 gigs is probably a safer estimate. Okay, so let's not worry about that for the moment. I've got some software that allows me to build these graphs. I can do humans. I don't need to worry about it. What can I do with it once I've got this data in a graph? I can look for these bubbles in a fast manner, and that's something, if anyone's interested, I'm happy to talk about afterwards, because it's quite surprising that we can find bubbles in a linear complexity algorithm. You'd expect any algorithm that had to sort of search a graph to be slower than that. But for the moment, let's just say we can find bubbles. And because the size of my graph depends only on the size of my genome, actually, once we've broken the memory barrier, we could actually put multiple people in at the same time. So each node contains the word that it represents, but also contains, for each individual in my data, both an edge, a bunch of edges, and a weight telling me how often it's been seen. And other things we can do with it, we can, so one of the interesting things you can do is, what you've done is you've built a de novo assembly of your data purely with respect to itself, and then afterwards you can align the reference to the data and compare how the reference relates to your data. And so instead of confounding your reads by trying to map them to the reference, you do it the other way around. So what does this mean practically? It means you could assemble E. coli on your iPhone if you really wanted to, and desperate to sort of stay connected with your work. You could do 100 E. coli on your laptop, 1,000 yeasts on a standard commodity server. But humans are still tough. You need a fairly big server, and I've given an example there of about 10 humans in 180 gigs of RAM. And the only way you can do that, because sequencing data does contain a lot of errors, is you build a binary graph of each person individually. You deal with sequencing errors. We have methods for error correction. And then, having built a clean binary, you then start assembling them all together into one multicolored graph. So what do I mean? What am I talking about with these graphs? Here's quite a busy picture. I'm only really going to talk about the top left. So what I've done is, this is just a cartoon of what you have in a graph, say, of a human in blue with the human genome reference in red. So if we look at this top left bubble, you have a bubble in blue, so you have a heterozygous site, and you have only one branch in red, which is the reference allele. If you look at the next bubble along, you've got a bubble created by using both colors, but not in either color alone. So you have a reference allele, but your individual is homozygous non-reference. And most interestingly, just above where it says figure 1A here, you've got a bubble which is there in both colors. And the key thing about that is, the only way you could ever see a bubble in red, which is your reference, is if it's a repeat. So what we've done is we've found a very simple way of disentangling repeats from polymorphism. All we do is we load the reference in, we look for all the bubbles that are there in red, and we decide we're going to ignore them. Then we look in blue and look for bubbles there, and we ignore the bubbles that were there in the reference already. And that sort of drops our false positive rate hugely. And there are some other examples of things that you might want to do when you've got multicolors. You could load in different genomes of different species to compare them. You could compare assemblies of different individuals at the same site, and you might use one of these individuals seems to have some gaps, but you might use the other individual to help them, and you might align the reference to the assembly. Okay, so this is all a little bit hazy. Maybe we can we model our sensitivity to finding variance? So the probability of detecting a variant is basically the probability that you have enough coverage, that there are reads that have been taken from that part of the molecule. It depends on the length of the variant, whether or not there are enough reads that cover it, times the probability that, given conditional on having enough coverage, the bubble itself isn't confounded. So if you've got a nice bubble, but actually other parts of the genome have come around and wrapped over it, in other words, it's a very repetitive region, then we're not going to be able to find the bubble, not with our current algorithm. So what we'd like to do is understand how these things work together. And now the top thing, the probability that you have enough coverage, is just standard Landau-Waterman style Poisson model. The only difference is it's on a De Broglie graph, so your depth of coverage has changed. But the thing that I want to talk about now is this probability that your bubble isn't confounded, assuming you have enough coverage. And really that's something that depends on the nature of your actual genome. So for different species, that would be different. And what you want to do is get an understanding of the species that you're interested in, and that will inform you about how your assembly is going to go. So let's do that for humans. Let's take a sample of SNPs. We'll take human chromosome 1, it's about 270 megabases. At each base on that chromosome, there are 3 possible SNPs. Let's consider all of the possible SNPs on the whole chromosome, and let's go through them one by one, add them onto my graph, and see if they overlap some other bit of the graph. And if they overlap some other bit of the graph, I'm stuck. It's not making a clean bubble, I'm not going to be able to see it. But if they don't, then that's fine. That's a bubble I expect to be able to see. And so the proportion of bubbles that don't overlap with the rest of the genome tells me something about the genome complexity. And in fact, it tells me something about single base errors, because a SNP and a single base error in some sense are the same thing. Now, so there's 2 things you want to look at here. First of all, let's look at the red curve. The red curve is essentially the thing that is the property of the genome, and that's showing the percentage of these new alleles created by new SNPs that don't overlap the rest of the genome. And at KMA 21, for example, only just over 60% of your bubbles are novel. In other words, more than 30% of sequencing errors at KMA 21 overlap other parts of the genome and confound you. And only when you start moving up to higher KMA levels do you start managing to avoid that. So, okay, so this is a bit theoretical. Maybe I should check it with some simulations. If I take my Poisson model for giving me enough coverage on my bubbles, and this thing, which tells me something about the conditional probability that my bubble is clean, given I have coverage, I can make some predictions about HOM and HET SNPs, about whether or not I'll be able to find them at different coverages. So these, on the left you have HOM SNPs, on the right you have HETs. Let's think about HOMs for a second. What are the properties of these curves? Essentially, as you go from left to right, the graph tends to go up at first, and that's driven by the curve we've just seen. Essentially, as you go from left to right, your bubbles are less likely to be confounded, and then your graph kind of collapses. And the reason for that is, as your k gets close to your read length, you're demanding almost complete overlap between your reads, and so that completely kills the effective coverage. So those are our predictions. We ran some simulations. We took diploid human chromosome 1 and ran at 10, 20, 30, 40, 50x coverage of 100 base reads, and 20, 40, 60, 80, 100x at 36 base reads. I'm just give you the results for just one of those. and the results match pretty closely what we predict. Okay, we're almost at the end of my first section where I just talk about what we can do, and I was going quite fast. But I wanted to give you a flavour of the kind of things that we can confidently do now with reference-free approaches. So there's a couple of obvious use cases for this kind of thing. First of all, let's think about that trick where we had a graph which had a red curve, which was your reference allele, and I used any bubbles in the reference allele to sort of distinguish repeats from polymorphisms. Why can't I do that with some species where I have a homozygote strain and a bunch of wild types? I want to distinguish between variants between my wild types, but I haven't got an assembly of them, but I have got a homozygote strain. So let's load the homozygote in at red, ignore any bubbles that are in that, because any bubble in that, effectively you've got something that's almost haploid, and any bubbles you see are going to be repeats. And then what drops out is differences between your wild types. And then you can play all kinds of games about things that segregate your wild types depending on what you know about them. And you can play a similar game with cancer match pairs between normal and cancerous. You just have the two in two separate colours. They're sequences from the same individual. So at almost all places, the graphs would be the same, and the distinctions would allow you to sort of pull out automatically variants that distinguish them. Okay, so that's the first half. Now the question is, what do we find hard? For an individual, there's obviously, if you really want to understand the genome of an individual, you need to understand the bits that are close to repeats. And what I've done so far is I've modelled what's our ability to sort of avoid repeats and call variants everywhere else, but really what we want to do when we want to get to know things well is we need to break into those repeated regions. And also, we want to start getting longer range information. If I know something about this individual at this this variant, and I know something at this variant, maybe I can start tying phase information in. And so we've got to work on the right data structures in order to be able to do that in our graph. And secondly, and this is the really tough one, is that thanks to people like Gil and Richard, we're going to have to deal with thousands of genomes. And I don't have memory to put them all in their own colour. I can do 10 people, but I certainly can't do 2,500. So what are we going to do? We want to understand populations. We think it's a good idea to try and look at all of the variation in the population at the same time. So the only choices we have are pool our data in per population and look at population graphs, or have a graph which has two colours, one colour which is sort of a pangenome of the whole population, another graph which is the individual, and let's look at inference about the individual in the context of the joint graph. So what are the kind of things that we need to worry about? Let's take a concrete example, because I think you need to see the kind of things that go wrong. If we take one of the HLA alleles in the MHC, they have a whole bunch of there are 1,400 in fact alleles of this gene. It's one of the most diverse genes in the genome. If I pick one of them, and if I just pick the region containing the exons and I draw a graph of it, obviously it's a whole load of nodes. Now I'm going to carry on drawing more and more complicated graphs, and in order to make them more comprehensible, if I have an arc of the graph which is completely unambiguous, I'll just squish it into one blob, and the blob will correspond to the size of the arc. So basically what I'm saying here is that just this part of the exon is completely unambiguous. No KMO is repeated, no part of this exon looks like any other part of the exon. That's great. What happens if I compare another allele? What are the kind of things that can happen? Okay, that looks exactly what you would expect, really. In the middle, these colours don't come out very well, but in the middle you have these shared KMOs or shared contigs, and on the outside you have the bits that distinguish the two alleles. So they come together and separate, and as the sequence sort of looks similar and then looks different. So let's go back for a minute to the first allele, which I just said was completely unambiguous. Now let's put it in context of about 80 kb of sequence around it. What does it then look like? Okay, it's a mess. What's this telling us? Well, it's telling us that lots of the sequence around the exons of this HLA allele actually looks like the sequence in the exons. And so you've got you've got this whole sequence here which seems to be almost repeated a second time, but with slightly different sequence. And now if I add the second allele that I just had before, it's even worse. And I haven't even, and the MHC has 3 paralogs, and many of the nodes in here actually occur hundreds of times elsewhere in the genome. So if I'm interested in looking at genomes in the context of the whole genome of the population, I need to be able to somehow cope with the complexity of this graph. Now, one way you could do it is just by increasing k, and it becomes very simple. But that's not practical because even though read lengths are increasing, actually the distances between our errors aren't increasing, and we never really want to have a k bigger than the mean difference between our errors. So let's stick to a smaller k for the moment, and let's keep thinking about this. What happens if we put in all 1,400 alleles of this HLA gene? It's insane. I must be mad thinking about this. I mean, what can we possibly do? So there's one big piece of information we haven't yet used, and that's read pair information. Okay, so now let's think about what kind of information could read pair information gain us. Now we know that basically read pairs consist of 2 reads, length r, and a gap of insert size of length I. So the amount of information inside 2 paired reads is strictly less than 1 massive read, which contains both of the reads and the insert. So for example, 200 base reads with an insert size of 300 is strictly less information than a 500 base read. So let's see what I could do with 500 base reads. If I can't do it with 500 base reads, I certainly can't do it with 2 100 base reads and an insert size. If I now look at this joint graph of all of these HLA alleles, what does it look like? It's still not much better. It's bluer, which on the color scheme basically means things are less repetitive, but I can't just use the topology of the graph and look for bubbles. Okay, it's still too complex. Okay, so what are my challenges? Despite what I've just said about read pairs not completely disentangling the joint graph, read pairs still give me a lot of information, and we need to look for new ways of. Read pairs tend to be used in a haploid context, where what you want to do is tie together contigs into sort of longer contigs. But what we want to do here is tie together bubbles, and you want to tie together bubbles in some manner that you can sort of reuse. So for example, if I have a person who has one bubble here and another bubble over here that's got a repeat on top of it, what I really want to do is use my read pair information to sort of separate these out so I can distinguish them, and separate them out in a manner that sits in RAM in a useful way so I can sort of traverse the graph and use it. That's difficult. And so that sort of covers points 1 and 2, and the final thing really is, I don't want to wait until read lengths are longer. I have plenty of information now. Can I perform inference on the graph even now? Even though it looks so horrible, maybe I can still work things out. So let's do that. Let's take a really tough part of the genome that's got considerable medical interest, and that's the HLA, which coincidentally I gave some examples of. And let's see what we can do. Let's see if we can examine the genotype of HLA-B, which has got this ridiculous number of alleles, 1,400. And let's take an individual from the 1,000 Genomes project, where we've got 25x of Illumina data and 8x of 454 data. And let's build a graph with loads of colors. One color is the individual, one color is the reference, and let's put one color for each of the different alleles. And then let's look at the graph let's look at the individual in the context of that massive graph. And what I want to do is go through, there's 1,400 choose 2, it's about 1 million different genotypes, and for each of those genotypes I want to calculate the probability that I would see the data I see. So I do that under a model I'm not really giving details of. This is very preliminary work, and the graph looks like this. So what do we have? We have the vast majority of our genotypes. So there's no meaningful way in which I can order these. So I've ordered the genotypes in order of likelihood. And so you can see immediately that most genotypes are extremely unlikely, and there's a small number at the left-hand side which seem to fit the data better. And if I zoom in, actually I only have one contender that seems to beat the others. And what's that? That matches the HLA typing that we have experimentally from the HapMap project, because this is a HapMap individual, and so they were HLA typed serologically. So it seems that we found a way to sort of use the information inside the joint graph to work out something very concrete about this individual despite the complexity. And I think that, I'm not sure if I'm well over time, I should think. So I should finish and say this is joint work with Gil and Mario. Thanks to the 1000 Genomes Project for data, and to conversations with Adam Orton and Stephen Leslie.